Picture a world where millions of identical, healthy plants spring to life without a single seed in sight. This isn't science fiction, it's the groundbreaking reality of plant tissue culture. Welcome to the Fields of Innovation YouTube channel, where smart farming starts here. Discover the future of agriculture. Today, we're diving deep into a technology that's revolutionizing how we grow plants, feed the world, and preserve our planet's biodiversity. Plant tissue culture is a cutting-edge laboratory technique that uses tiny pieces of plant tissue, called explants, to regenerate entire plants under sterile and controlled conditions. As global demand for food, sustainability, and plant-based resources skyrockets, this technology offers a game-changing solution for mass-producing crops, ornamental plants, and even medicinal species with unparalleled efficiency. But what exactly is plant tissue culture? At its core, it is a set of techniques that allow scientists to grow new plants from small pieces of existing ones, all within the controlled environment of a laboratory. The key to this marvel lies in a concept called totipotency. Every plant cell has the remarkable ability to regenerate into a whole plant when given the right nutrients and environment. This scientific principle forms the foundation of tissue culture and opens up a world of possibilities. The idea of plant tissue culture isn't new, it dates back to the early 20th century. With pioneering work by scientists like Haverland, over the decades, this technology has evolved into an indispensable tool for agriculture, horticulture, and biotechnology, pushing the boundaries of what's possible in plant propagation. Let's walk through the step-by-step process of plant tissue culture. It all begins with selecting an explant, a small tissue sample chosen from a parent plant, often from leaves, stems, or meristematic regions. This tiny piece of plant material holds the potential for an entire new generation of plants. Next comes the crucial sterilization process. The explant is carefully cleaned to eliminate any bacteria or fungi, as even the slightest contamination can ruin the entire culture. Once sterilized, the explant is placed in a nutrient-rich medium containing sugars, vitamins, and plant hormones such as auxins and cytokinins. This special cocktail provides everything the plant cells need to thrive and multiply. Under the influence of these hormones, the cells begin to divide rapidly, forming an undifferentiated mass called callus. With the right hormonal balance, shoots start to emerge from this callus, marking the beginning of new plant growth. Once the shoots develop, they're induced to form roots, creating complete plantlets. But the journey doesn't end there. These delicate new plants need to be gradually acclimatized to conditions outside the lab. It's a careful process of hardening off, preparing them for life in the real world. Finally, the young plants are transferred to soil or greenhouse conditions, where they grow into fully functional plants, ready for agriculture or horticulture. The advantages of plant tissue culture are numerous and far-reaching. One of the most significant benefits is the production of disease-free plants. Because they're grown in sterile conditions, plants produced through tissue culture are free from viruses and pathogens, ensuring healthier crops and higher yields. The speed of multiplication is another game changer. Millions of genetically identical clones can be produced in a short time, making it ideal for large-scale farming operations that need to quickly ramp up production. This rapid multiplication also plays a crucial role in conservation efforts, helping to preserve endangered plant species and maintain biodiversity. Uniformity is another key advantage. Since all plants are clones, they grow with the same characteristics, ensuring consistent yield and quality, a huge benefit for farmers and consumers alike. And unlike traditional farming methods, tissue culture isn't bound by seasons or climate, offering the possibility of year-round production. The real-world applications of plant tissue culture are vast and varied. In agriculture, it's widely used in producing staple crops like bananas, potatoes, and sugarcane. The horticulture industry relies heavily on tissue culture for propagating orchids and other ornamental flowers that are in high commercial demand. Forestry is another field benefiting from this technology, using tissue culture to support reforestation efforts by producing fast-growing trees like teak and eucalyptus. In the pharmaceutical and biotech industries, tissue culture is essential for growing medicinal plants that provide raw materials for life-saving drugs. The global impact of plant tissue culture is profound. Countries like the USA, Canada, UK, and Australia are applying this technology in large-scale farming, horticultural exports, and sustainable forestry practices. It's playing a crucial role in addressing food security challenges by producing high-yield, disease-free crops that can help feed growing populations around the world. Moreover, tissue culture contributes significantly to sustainable agriculture by reducing dependency on chemical pesticides, minimizing land use, and enhancing biodiversity conservation. It's helping to create a more environmentally friendly approach to farming. Looking to the future, the potential for plant tissue culture is even more exciting. The use of automation and bioreactors could revolutionize mass plant production, dramatically reducing costs and increasing efficiency. Imagine vast facilities where millions of plants are grown in perfectly controlled conditions, with minimal human intervention. However, challenges remain. The initial investment and need for skilled labor are significant hurdles, especially in developing regions where this technology could have the most impact. Overcoming these obstacles will be crucial for the widespread adoption of plant tissue culture techniques. The integration of artificial intelligence, robotics, and smart farming systems with tissue culture techniques holds promise for further optimizing crop production. We might soon see AI-driven systems that can predict the perfect conditions for each plant variety, or robots that can handle the delicate process of transferring plantlets with unprecedented precision. In conclusion, plant tissue culture is truly a groundbreaking technology that allows for the mass production of plants with unmatched speed, quality, and consistency. It's transforming agriculture, horticulture, and conservation efforts around the globe, offering solutions to some of our most pressing challenges in food security and environmental preservation. We'd love to hear your thoughts on this fascinating technology. Have you seen plant tissue culture in action? Do you think it could revolutionize farming in your area? Leave your comments below, and don't forget to subscribe for more insights into agricultural and biotechnology innovations. The future of farming is here, and it's more exciting than ever. Until next time, keep innovating in your own fields of discovery.